Hello, everyone. My name is Veronica Hernandez. I'm a student at the Dr. Kaizing Lab through the Department of Biological Sciences here at Texas Tech University. And today I'll be presenting my research over ergosterol synthesis and how it affects nutrient uptake in Leishmania major. So starting off, Leishmania major causes a disease known as leishmaniasis, and it causes this cutaneous form which appears as ulcers and permanent scarring on the skin. This can lead to a reduction in the quality of life as individuals can unfortunately accumulate a great number of these scars as they are exposed to the disease. Our current medications and treatment options are inadequate. They cause unfortunate side effects such as nausea, vomiting, and those medication treatments are also expensive and difficult for individuals in third world countries to actually receive those treatments. And to go along with that, there's also no vaccine for this disease. So we are hoping by understanding the basic biological functions of Leishmania major, we can facilitate drug development and eventually one day even a vaccine. So the global impact of leishmaniasis. This shows just the cutaneous form of cases from 2019. And we see most of the cases in the Southern American regions, the upper north portions of Africa, and the Middle East, with up to 1 million cases per year, just to further show that this is a pressing issue. It affects many people worldwide, and we are hoping to reduce the impact of this very unfortunate disease. So the Leishmania life cycle. Leishmania is known to go through what is known as a digenetic life cycle, otherwise that goes through two different hosts throughout its lifetime. The first being the sandfly. When it's in the sandfly, it survives as a promastigote form, and this is a more slender, motile form of the cell. And afterwards, it turns into an amastigote form when it enters the mammalian host, otherwise human beings in this case. When it enters the host, it is accumulated in macrophages where it then multiplies and leads to those ulcers, as we can see there on the bottom corner. So whenever Leishmania promastigotes are surviving in the midgut of the sandfly, it endures many different challenges, such as competing for nutrients with other microbiota, bacterial toxins in its environment, and even osmotic changes during sugar and blood feedings. So Leishmania cells have to endure these stressors in order to eventually form into the amastigote form within the mammalian host. So sterols. They're a crucial membrane component in Leishmania and a very crucial component in lipid drafts. About 30% of Leishmania's membrane is made of sterols, and these lipid rafts play a key component in membrane functions such as stress response and signaling. And what's so special about ergosterol and sterols is that Leishmania synthesizes ergosterol, unlike mammalian cells, which synthesize cholesterol. So this is a very promising drug target, as we can hopefully utilize this to destroy Leishmania without actually hindering the individual. And so what mutant we use to observe this component is the C14DM mutant, C14 being C14 demethylase. This mutant lacks the key enzyme in ergosterol synthesis. So by using this mutant, we can see the effects and the importance of ergosterol in its cell functions. So some previous results with the C14 and SPT2 mutants, again, C14 being the C14 demethylase sterol mutant, and SPT2 being the serine palmitotransferase 2 sphingolipid mutant. Sphingolipids also being an important part of the membrane, but roughly making about 10% in comparison to the higher 30% of sterols. First, we place these different cells in PBS, being a media that does not have any nutrients. So we tried to mimic starvation conditions. And in doing so, we saw that both mutants had a much higher death percentage rate in comparison to the wild type and add-back cells, with add-back being cells who had certain genetic material removed and then added back once more, and they are expected to behave like wild type cells, as we can see here. However, despite that higher percentage death rate, once we were added glucose into that starvation media, we were able to rescue those cells so that they no longer had such a high death percentage rate. But nevertheless, we were able to see that both of these mutants have hypersensitivity under starvation conditions. So further previous results, just focusing on the C14 sterol mutant, we had exposed them to Triton X and viewed them under microscopy. So that first set on the top images are wild type mutant and add back cells that did not have this Triton X agent added, Triton X being a membrane perturbing agent. Versus on the bottom, that red color you see is the Triton X that has been taken up by the cells. So that mutant had taken up much more Triton X in comparison to the wild type and C14 add backs. And just to provide a little graphic, these normal membranes were able to withstand the presence of Triton X versus this mutant had a compromised membrane in that Triton X was able to enter and hinder the cell. So objectives of this project: how does the synthesis of ergosterol and sphingolipids protect leishmania during glucose starvation? And does ergosterol and sphingolipid imbalance alter glucose uptake and consumption during starvation stress? And our hypothesis to go along with that was sterile and sphingolipid mutants will incorporate more glucose and increase glycolysis in order to compensate for their defects in respiration. So basically that they're going to take in much more nutrients due to the fact that they've been compromised and that they cannot synthesize ATP at a regular rate in comparison to their wild type cells. And the way we did this is we gathered some permastogout cells. They were washed to get rid of the media that they were grown in. And we layered a tube starting out with dibutylphthalate oil. And on top of that, a layer of aqueous PBS radio labeled with C14 glucose. And in doing so, we were able to incubate the tubes over a period of time and terminated that incubation with high-speed centrifugation so that whenever they were centrifuged, only the cells that uptook glucose were pulled down into that oil layer, and the glucose that was not utilized by the cells remained in that top PBS layer. So by doing that, we were able to flash freeze these tubes and snap off those tips to analyze with liquid scintillation counting due to that radio-labeled glucose. And before we went into the main parts of our experiment, we just did basic background check with just working with wild type cells under room temperature and 4 degrees Celsius. And the reason we did this is just to confirm what we already know, which is that glucose is an active transporter and does not function well under low temperatures. And it behaved exactly as we expected, with the 4-degree exposed cells having little to no uptake of glucose versus cells at room temperature incorporated glucose steadily at a greater rate over time. So once we put these cells under starvation conditions, they all had roughly the same trend, as in over time, they all incorporated a greater amount of glucose. But what we could see most notably is that the C14 sterile mutant uptook a much greater amount of glucose in comparison to any other type of cell, most likely to compensate for the fact that its membrane has been compromised and that it is unable to function at a regular rate in comparison to other cells that do not have that hindered membrane. So conclusion: sterols protect the mitochondrial function of Leishmania major during glucose starvation, and ergosterol mutants, the C14 mutant, significantly take up more glucose than their wild-type counterparts, possibly as a mechanism to adapt to overcome their respiration defects. So future directions we would like to take this project is to determine the role of sterols and sphingolipids in incorporating other small nutrients such as glycerol that are relied on the cells during gluconeogenesis in the absence of glucose or in starvation conditions. And another direction we'd like to take this is to determine the relationship between these glucose transporters and drug uptake in various species of Leishmania, just trying to see the connect of how does a greater incorporation of glucose translate to the uptake of drugs, the treatments that are used on Leishmania today. And just to close out, special thanks to the Dr. Kai Zhang Lab, the Department of Biological Sciences here at Texas Tech University, and the Honors College Undergraduate Research Scholars Program supported by the C.H. and Helen Jones Foundation. Thank you.